My name is Lien Trinh. I'm an optometrist based on Larrakia country in the Northern Territory of Australia. You're listening to Talking Eyes. So this episode is an introduction to a very special bit of tissue at the front of the eye, the cornea. And I've had many patients coming through my consulting room over the years. And when I've mentioned it to them, and when I've discussed the cornea with them, you can sort of see that their faces are wondering, is it the coloured part? Is it that white part? Is it the black part? Well, today we will be distilling exactly which part of the eye it is. And to help me, I have one of the brilliant chief scientists at the Centre for Eye Research Australia who has been working on this little bit of tissue for over a decade. Dr. Karl David Brown, welcome.
Oh, thank you.
Now, Dr. Karl, actually, I should mention that Dr. Karl was one of my inspirations for starting this podcast, and I listen to you all the time, Dr. Karl. So if you're out there listening. And so I'm so thrilled to be hosting a Dr. Karl podcast today. But how did you end up in corneal research? Because you're a scientist by trade, aren't you? Like a straight old-fashioned scientist.
I don't think I'm that old-fashioned. So I had a long-standing interest in regenerative medicine and tissue engineering. And
from what age? Like, were you dreaming about it as a kid? Is this something that...
From about 25.
Right.
And sort of as I learned more about biology and the potential for the technologies that were coming down the line, I wanted to get involved. And the cornea was a really interesting place to start for that kind of work. And I'm sure we'll go into this in a minute, but It's relatively simple structure. You can see it, you can monitor it. It's on the outside of the body, et cetera, et cetera.
I mean, to a lay person, it looks fairly basic. And throughout optometry school, when I was studying the cornea, I thought it was fairly basic as well.
I only do easy stuff.
But it's not. As we're learning more and more, it is very unbasic.
Yeah. I mean, it's the clear part at the window, if you like, to the eye. And despite being, say, half a millimetre thick, it's got 5 layers. So starting on the outside, facing the air, is the epithelium, which is kind of like the skin of the cornea. And then there's a Bowman's membrane, which is just a protein membrane that the epithelium sits on. And then a spongy layer called the stroma that makes up most of the thickness. And then the Descemet's membrane, which is just another membrane of protein. And then on the outside of the Descemet's membrane, facing the inside of the eye, is a layer 1 cell thick called the corneal endothelium. And that's mostly where I do my work.
At the corneal endothelium?
At the corneal endothelium. It's 1 cell thick, it's easy.
So not only have you specialised in this tissue of the body, but you've actually subspecialised. And so let's talk about the cornea itself, because it does serve a number of different functions within the body.
So the cornea is on the outer surface of the eye, and it's obviously transparent. Otherwise, you can't see. And when it's not transparent anymore, that's a problem. It protects the rest of the eye, like the iris and the pupil and everything else, from damage.
It's really strong, isn't it?
It's very strong.
Yeah.
So it works as a physical barrier. It also stops infections from getting into the rest of the eye, etc. The other thing that a lot of people don't realise, though, is it actually helps focus the light. So it's curved, and that bends the light towards the lens, and then the lens does the rest of the focusing. So if your cornea gets to be the wrong shape, for example, there's a condition called keratoconus where it gets sort of more cone-shaped, then that upsets that focusing and degrades your vision.
One of the things I think we take for granted is its transparency. We sort of just, think, yes, that's what it is, that's what it does. And then all of a sudden, when it is no longer transparent, we realise there's actually a whole bunch of processes that are going on that maintain that transparency. What is happening?
So, the cornea is not like just a piece of glass that's transparent all the way through. There are a lot of different things that are happening to keep what is a piece of bodily tissue transparent. So the epithelial cells don't have much pigment, so that makes them transparent. The stroma is probably the most important thing to think about. So the stroma is that spongy layer in the middle, and it's actually made up of layers of fibers, all very carefully aligned. And so one layer will go one way and the other layer will go the other way, and they're all at very precise distances apart from each other. And so as the light goes through, you get interference, and the end result is this transparency. But if those distances get messed up, if the cornea swells, then light scatters as it passes through the stroma. The cornea looks white, and then you can't see because you can't see through it.
You talked about the fibres within the stroma, but the bulk of the stroma is made up of fluid, isn't it?
Yeah, so the stroma has a fluid content, and one of the way that those fibres can get out of alignment is if fluid goes into the cornea, too much fluid, and it swells up.
How does fluid get in?
So now we get into my favourite bit of the cornea, the corneal endothelium. So the endothelium maintains that fluid balance. So it has two things. It has a leaky barrier. So fluid will flow in from the inside of the eye into the cornea, but slowly because it's a barrier, but it's leaking. And
on purpose.
On purpose.
Yes.
And the second thing is that's balanced against a pump function. So the corneal endothelial cells actively pump fluid out of the cornea. And between those two, you get the Goldilocks effect of just exactly the right amount of fluid in the stroma. So if the corneal endothelium gets damaged, then a couple of different things can happen. First is that barrier function can be disrupted and you can get more fluid going in, and or the pump function slows down or stops, in which case you're not getting enough fluid pumped out, and that balance goes out of whack. Fluid builds up inside the cornea swells, the distances between those fibers gets disrupted, they get all buckled, and when light hits the cornea, it hits the fibers and scatters, and the cornea looks white. So one of the things we can do nowadays is just replace the corneal endothelium with donor tissue. So instead of having the entire cornea replaced, you can just replace the corneal endothelium on that Bowman's membrane, the protein membrane I mentioned earlier, and that'll restore the pump function, and the fluid will go back to normal, and the cornea will go from white to transparent again.
Wow. With minimal disruption to those collagen fibrils. That's amazing. Mm-hmm. I suspected there might be some sort of floorboard effect where, when your floorboards become swollen, when they dry out again, but it doesn't work that way. No, the cornea is amazing in that sense. They do
fall back into position unless they get scarred, which is a whole other process.
Right. And so what happens with scarring then?
So with scarring, the immune system gets in and starts to remodel the fibers or put extra proteins in. And so that's an extra disruption to that, to all that careful spacing that you don't see if it's just swelling. So if it's scarring, then you have to do a full thickness cornea transplant or replace the stroma and the epithelium.
Let's step back to these pumps, these corneal endothelial pumps, and talk about what the purpose of the pumping mechanism is, because why leak fluid into the cornea and then pump it back out? What role does that play?
Yeah, I mean, why not just have like a clear piece of glass at the front of your eye?
Exactly.
It seems easier. So the cornea is a living tissue, and little scratches and things it can heal, usually as long as the stroma isn't damaged. So it's alive, it grows with you, and because of that, it's made up of cells and fibres and all the things that we've talked about already.
Yeah.
And cells need oxygen and nutrients and all those things. So this flow of fluid lets those nutrients in, but it also lets an exchange of the waste products of metabolism out.
I guess when you think of nourishment of bodily tissues, you're always thinking about blood vessels, aren't you? So you're thinking about blood flow, bringing oxygenated blood into the area, removing deoxygenated blood away from the area. But that doesn't happen for the cornea.
And I think that's one of the reasons why the cornea is so good for tissue engineering, right? Because one of the big problems with tissue engineering is getting those blood vessels in, you know, if you're going to make a kidney or something. But the cornea doesn't have them, so that's easy. All you got to do is...
Did you hear that, all those corneal tissue engineers out there? This is easy.
Easy. No, so the cornea gets its oxygen from the air and from the aqueous humor. And so there's, you know, the fluid in the front chamber of the eye. And because it's so thin and because you've got this interaction with fluid flowing in and out already, it doesn't need the blood vessels.
So you've got nutrients coming in behind, nourishing the endothelium. And where are the nutrients coming from to nourish the epithelium on the front surface?
Yes.
Is that happening?
Yeah, so I mean, you've got fluid exchange through the stroma to the epithelium, and you've also got oxygen absorption through the tear film.
And the interesting thing is that the epithelium is constantly regenerating, isn't it? How does that happen?
Okay, like your skin, there are these adult stem cells that are on the very edge of the cornea, where the cornea meets the sclera, the white of your eye. And those adult stem cells divide, and when they divide, some of them go on to slowly develop into more corneal epithelium, which sort of migrates from the edge towards the centre and then up and out. And so you've got this constant turnover, slow turnover of corneal epithelial cells. One of the things that can happen, though, is if those adult stem cells on the very edge of the cornea where it meets the white, if those are knocked out because, you know, of a terrible burn or something like that, then you're not having that source for new corneal epithelium. And when that happens, the epithelium doesn't regenerate and you end up with scar tissue.
So you're talking about that area where the sclera, the white part of the eye, meets the cornea, an area that we call the limbus, where all the stem cells are located. Tell me about these stem cells, because, I mean, are they going constant duplication to constantly replenish that surface?
Yeah, so the stem cells divide generally very slowly, but they produce transient amplifying cells, which is like the next step towards becoming a corneal epithelial cells. And they divide, and as the cells divide and grow, they're also migrating towards the centre and up and out. So if you can imagine like sort of a spiral.
They're spiralling in towards the centre of the...
and upwards. So, and then by doing that, they're constantly renewing the epithelium, which is several cell layers thick. And if the stem cells are completely knocked out, then you don't get that renewal anymore.
You don't get that renewal.
So the endothelium, the rate that those cells divide is very, very slow.
Right.
So traditionally in the old textbooks, they would tell you that they didn't divide at all.
Yes, correct.
Nowadays we understand that they can divide, but that rate of division is so slow that it can't keep up with just the wear and tear of being alive. And the numbers go down as you get older and older. This means that if you have damage to that inner layer, then it doesn't repair by itself, and you're looking at a cornea transplant.
Yeah. And the endothelium is a single cell layer.
It's one layer, cell layer thick.
And it's doing a lot of work.
It's very metabolically active. It's very active, and it's doing that pump function, which it, keeps the rest of the cornea clear. So it may only be 1 cell thick, but it's a very important layer.
I recently learned that the body's immune response in the cornea might largely come from the corneal stroma.
Mm-hmm. The Langerhans cells are kind of like the immune cells of the cornea, and they live in the stroma. And so when we do cornea transplants, if you do a full thickness transplant, you're transplanting the donor immune cells as well as the stromal cells and the epithelial cells and the
everything else
cells,
that you want.
Everything else.
Yeah.
Whereas nowadays, often what they do instead is just replace the damaged layer. And frequently, the layer that's damaged is the endothelium. So if you're just replacing the endothelium, you're not bringing those donor cells in. And we think that's why when you just transplant the endothelium, the rejection rates are much lower.
That makes sense. It's actually quite difficult to talk about the cornea and maintaining the integrity of the cornea without alluding to the structures that surround the cornea.
Mm-hmm.
You know, you've got your eyelids, which are really essential for maintaining the corneal integrity, maintaining its moisture, ensuring that front surface of the eye, the corneal epithelium, and I know you said you weren't the epithelial guy, but I'm sure you have some idea of the epithelium. Yeah. But I do recall a really, really painful experience I had as a young child, and it was the first time that I'd gone skiing, and I'd gone skiing with my older brother, and we happened to pick up some really cool blue glasses from the Coburg Trash and Treasure markets back when I was 14, and I thought these will be perfect to go hit Mount Buller with. And I spent the day skiing in these blue glasses from the Trash and Treasure market, and I came home that night just with the most painful, gritty, sore, watery eyes, and it just felt like I had sand in my eyes for days. Yeah. What happened?
It's kind of like sunburn.
It's sunburn,
it's
essentially.
Basically sunburn on your eye. And so, you know, the recommendation would be wear actual sunglasses.
Good UV protection, essentially. Yeah. Because when you are out in the snow, the dosage of UV is huge, isn't it?
And snow blindness has been a known problem for a long time. So at the corneal level, the UV is getting in and essentially burning the same way that you would get sunburned.
I mean, when I'm looking at a cornea on the slit lamp that has had UV burn, it's looking very sad. It's looking very punctate, I guess, is the clinical term for it. So there's lots of spots on there, lots of areas which show positive staining. So when we stain the cornea, we use fluorescein in clinical practice.
And you get those little glowing spots.
We've got lots of glowing spots which indicate that cells have died. Correct, But we talked about how amazing it is that the cornea can regenerate its epithelium. So the epithelium can regenerate. The cornea's stroma, can that regenerate?
So the cornea's stroma, the cells can divide, but the stroma has all those collagen fibres in it, which are very carefully aligned. And once those get damaged and you get remodelling, the cells go in and sort of try to fix it up, but they don't do a great job.
No.
At that point, all those alignments go out and you get cornea scarring. The only recourse is to transplant either full thickness, so replacing all 5 layers, or an anterior transplant, so just replacing all or part of the stroma and the epithelium.
So apart from stromal edema, where there's been swelling of the stroma, it will not be able to maintain its transparency, basically, if there's been an infection or a traumatic injury, I guess, to the...
If the infection is at the point where you're getting that remodelling of the proteins that make up the stroma, then it's very hard to come back from that, and you're probably going to need a transplant.
Let's talk about the nerves, because the cornea is one of the most innervated tissues in the body. I mean, it's a really sensitive part of the body. Yeah. every time I attempt to put a new contact lens into a person, their immediate response really is one of flinching. They're terrified, but they really want it because it's life-changing. And that's an automated response, really. It's the response that we've evolved to have because we need to protect.
Get the eyelids over the top, protect the cornea.
Absolutely. Yeah. But it is a really sensitive part of the eye. Where are all our corneal nerves located?
Yeah, so there's a plexus of nerves under the epithelium. And one of the interesting things is that those nerves are obviously cut when you do a transplant, and then they have to regrow back in again, particularly if you do a stroma or full thickness transplant. And yet they do. So there is some capacity for regeneration there.
Of those corneal nerves. Let's talk about some of your work that you've been ploughing away at. What was your PhD on?
So my PhD was on corneal tissue engineering. And I looked at the corneal epithelium and the corneal endothelium during that work and trying to find ways of growing the cells in the lab and then arranging them on materials so they can be used for transplantation. And
so the epithelium is important when there's been a burden, like you say, and there's no regenerative capacity from the person who's experienced that injury. In practice, have we gotten to a point where we actually do that?
Yeah, so for limbal stem cell deficiency, and that's where those stem cells, the population of them falls below a threshold and they no longer are able to provide enough cells to maintain the surface of the epithelium. So when that happens, we can do a stem cell transplant nowadays. Generally, that's done in a couple of different ways. The first is to grow the cells as a sheet in the lab and then transplant that sheet onto the surface of the eye. And the second is to take little bits of tissue that have those cells in and essentially cut off the scar or remove the scar and then glue those little bits of tissue on and they will grow out from there. So the source of those cells, there's a few different sources you could use. If the patient has a good eye and a bad eye, you might be able to take a very, very small amount from the good eye and use that to seed those critical cells back into the damaged eye. You could use donor tissue, or interestingly, another thing that we do is take tiny biopsy on the inside of the lip and use that as a source of cells for those procedures.
Why the inside of the lip?
There are oral mucosa epithelial cells that have the same sort of similar sorts of stem cells in them. Right. And so they can sort of be used as a replacement source to rebuild that stratified, layered skin, if you like, that is the epithelium. My current work is on the corneal endothelium. And so what we're looking at is taking a hydrogel film, which is a carrier for the cells, so a thin sheet of this special plastic, and then taking cells from a corneal donor, growing them in the lab on multiple hydrogel films. Mm-hmm. And so instead of one donor eye treating one patient, instead you'll be able to take one donor eye, take the cells from that donor, grow it on multiple hydrogel films, and then implant those and treat multiple patients from each donor.
Wow. And so creating essentially like a little manufacturing plant of corneal endothelial cells.
Culture and grow them in the lab. And
how long does it take to culture and grow?
It's about a 14-day process.
And how long are they viable for?
So the corneal endothelium, so we're dealing with normal human corneal endothelial cells, and corneal endothelial cells in eye banks here in Australia can be kept for up to 30 days at room temperature in a special medium that the eye banks use.
And then where do these go? Because is the intent for them to be distributed to every ophthalmologist who requires an endothelium for their surgical?
Yeah, so We are in the process of figuring out how to mass produce these things. And the idea would be hopefully to distribute them through existing eye banks, through existing networks. Why reinvent the wheel? And then the eye bank would be able to treat them pretty much the same as normal donor tissue. And then the surgeon will be able to implant it as if it was normal donor tissue as well. But there will just be a lot more of it, because instead of being one donor, one patient, one donor, multiple patients.
Yeah. Because let's talk through the donor issue. I mean, Australia doesn't really have a donor issue. I think we've got enough corneal donors for the number of transplants that we need, but it is an issue internationally that we don't have this. So the work that you're doing is really important.
Yeah. And, and there's something like 13 million people waiting for a transplant worldwide, and only about 1 in 70 people who needs a cornea transplant has access to one. So this is a big problem, and part of the problem is tissue supply. As you alluded to, in Australia, the Australian public is very generous, and we have sufficient tissue for site-restoring transplantation. In other nations, that's not always the case, and you need a lot of resources and infrastructure to collect, store, and distribute that tissue to where it's needed.
Thank you to Dr. Karl Brown for taking us on our crash course into the cornea. Today, we've explored what the cornea is and discovered just how complex this seemingly simple window to the eye really is. Our understanding of its different layers is constantly evolving, showing us not only how much there is still to learn, but also the exciting possibilities that lie ahead. Thanks for joining me this season to talk cornea. There'll be a few extra resources linked in the show notes. I hope you found today's conversation valuable. If you enjoyed the episode, please like, follow and share my socials and the podcast with anyone you think might benefit. For my eye care colleagues, I've produced a special edition educational podcast for you, which will be available exclusively through the Optometry Australia and My Vision platforms. It's where you'll find a lot more technical jargon, but real clinical pearls to complement the season. Thanks for listening. This is Talking Eyes, and I'm your host, Lien Trinh.